Hey guys. All right. This is Unit 6, Topics 5 and 6. And this is actually like a really cool topic for me to be talking about. I really enjoy um, talking about gene expression or the regulation of gene expression. When I was a kid and when I was in college, I really wanted to do genetic research, and it kind of led me to a fascination in genetics and cancer. And this is sort of the whole idea behind it. So I'm excited. Let's get started. So, as far as gene expression goes, eukaryotes and prokaryotes alike have to be able to turn off and on genes at any given time. You can see this, the simplest example of this is insulin production in diabetic patients. Or I should even say healthy patients, right? So you eat something that has sugar in it, that glucose increases in your blood, and your pancreas goes on to the cells in your pancreas go on to produce the protein insulin, which then lowers your blood sugar levels, but then we don't need it anymore. We got to turn it off, right? So genes can be turned on and off based on environmental and internal cues, similar to like blood glucose levels. On off refers to whether or not transcription will take place. If transcription takes place, we then have an mRNA molecule. mRNA molecule gets transcribed, translation gets translated on the ribosome to become a polypeptide and then protein. So, on off refers to whether or not that first step will happen. And this allows cells to be super hyper specialized in what they do. It's really interesting. So let's talk bacterial gene expression. It's a little bit simpler. So bacterial gene expression works with operons. I'm just gonna put this on. Let's stop here. An operon is a group of genes, right? So we're talking on the DNA. Genes are coding regions. They can be turned on or off. They have three parts to them. There's the promoter, right? This is where the RNA polymerase can attach. Remember, bacterial and prokaryotic DNA doesn't need any transcription factors or anything like that. So we just have this promoter region. We have the operator, just kind of the on-off switch. We'll talk about that later. And then the genes themselves. So they code for related enzymes and pathways. They could be repressible or inducible, meaning repressible on to off. So transcription is usually on, but can be repressed, so it can be stopped. Or inducible, off to on. So when we say an enzyme or a protein, the rate of protein synthesis is repressed, or we can induce protein synthesis that is going from on to off based on sort of these sections of genes, these groups of genes that hang out together. It'll be a little more clear when we see it on the next slide. Pause here if you need to take notes. So the genes, uh, the genes need a regulatory sequence. So the regulatory genes produce a repressor protein that binds to the operator to block RNA polymerase from transcribing that gene. So it's always expressed at low levels, but the binding of a repressor to an operator is reversible. We're going to go through some examples, but it's essentially a gene that produces the protein that causes something to get shut off. Okay, so all of this stuff comes from DNA in general, but you have this repressor protein that is created from the regulatory gene sequence, similar to like rRNA, the pirate RNA. It's the RNA that folds and makes the ribosomes, right? This is the gene that produces the repressor protein. It's a regulatory gene sequence. We're going to see some examples. This may look familiar. Oh, sorry, go back. Here we go. Pause if you need to take notes. All right, here we go. So allosteric regulation. So this may look familiar to you from Unit 3. We talked about enzymes and allosteric inhibition of enzymes. This works relatively the same way. Allosteric activator, it is a substrate. You can see here, this is in its inactive form. Boop, 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 boop. And substrate over here will stabilize the shape of the enzyme so that the active sites themselves remain open. So we remember this from Unit 3, allosteric activator, or an allosteric inhibitor, right? So normally this is open in its active form. You can see open. And the allosteric inhibitor comes in over here, and the enzyme changes shape. Remember, change the shape, change the function, save the cheerleader, save the world, and it becomes inactive. So let's talk about repressible. So this was all, I'm sorry, I didn't mean to pause here if you got to take notes. So this is only one form. The allosteric regulation is only one form of regulation of gene expression. Either we're going to change how the enzymes that that help with this gene expression, or we're going to change the genes themselves or how they operate. We'll see that on the next slide. Cool. So let's first talk about repressible operons. Repressible operons, remember, are the ones that we are going from on to off. And an example of this is called the trip operon, TRP. It's the trip operon. It's in bacteria. It controls the synthesis of tryptophan. Since it's repressible, transcription's always active. So it's always on this synthesis to produce this protein. Tryptophan is always on. We're just producing more and more of it. So transcription's active, but it can be switched off by a trip repressor. Remember, we're going to shut it off. We want to shut it off. Allosteric enzyme that's only active when tryptophan binds to it. So when there's too much tryptophan built up in bacteria, the tryptophan will most likely bind to the repressor, turning it active, which then shuts off transcription for tryptophan. So it essentially becomes its own regulation. So we have the trip operon's always active. When there's a lot of it, when it builds up, it's used as a repressor. It essentially allosterically binds, which shuts off transcription. So no more tryptophan. We saw this again in the enzyme pathway as well. But here's kind of what it looks like. So these are genes, okay? Trip E, Trip D, Trip C, those are just gene sequences. You have your RNA polymerase here. That's going to try to go this way, okay? Normally, well, when there's too much tryptophan, the tryptophan here will bind to the repressor. The repressor kind of gets stuck over here to the operator, and RNA polymerase is blocked from transcribing more tryptophan. Okay, when there's not enough tryptophan or in the absence of tryptophan, it's not going to come and bind over here anymore. The repressor sequence, or the repressor, is not hanging out on the operator, and RNA polymerase can go over here. It's a physical block, right? We can't. RNA polymerase wants to go over here to transcribe this stuff, but it can't because this is sticky on here. All right, but when there's no tryptophan, this thing falls off. It's actually, again, kind of elegant, right? If we need more, it falls off and makes more. If we have a lot, we don't need it, it stops. And what this is we call this an operon, this whole sort of process, this whole thing. Pretty cool stuff. All right. Inducible operons, so things that go that are off that we need on. So an example here is the lac operon. So it controls the synthesis of lactase. It's an enzyme that digests lactose, the sugar that's in milk. Since it's inducible, so transcription's normally off. So we're not always producing lactase. The lac repressor is bound to the operator. We're going to see this on the next slide. It is allosterically active, meaning that it's bound to the operator and is active when the allosteric site is bound. So the inducer, I remember this from college. The inducer for the lac repressor is called allolactose. When it's present, it'll bind to the lac repressor, turn the lac repressor off. So this is all nonsense. Let me show it to you in in image form. Genes can now be transcribed. Here we go. So this makes more sense. So we have an operator here. Okay, there's a repressor that's on there, and it's sticky on there. All right, lac repressor is active and bound to the operator. So this is the repressor in its active form. You can see RNA polymerase can't go up here. And this is weird. That's in like a different language. Polymerase. Anyway, I love how everything else is pretty much the same. So you have a repressor over here that is active. It's bound to the operator. RNA polymerase can't bind. We can't make this lactase over here. But we have allolactase, right? That will bind somewhere else. This is the allosteric site, okay? Because this site up here, this is like the meat and potatoes site, hangs out with the operator, right? But allolactase, it'll bind, and this will become active. The whole lac repressor will become active. Polymerase will come and bind, and then go down and produce lactase to help us digest that milk. Okay, so it's kind of the opposite of the trip operon, right? Is that when the allolactase binds, the repressor falls off, as opposed to the trip operon that when tryptophan is bound, it's on, and then without it, it falls off. So it's just sort of the opposite. All right, quick check. Number 1, compare and contrast bacterial lac operon to the trip operon. Both regulate genes. Trip operon's repressible, meaning that it's going from on to off. Lac operon is inducible, going from off to on. What are the 3 parts of an operon? Promoter, operator, and the genes. All right, pause here, take notes, etc. All right, well, that's all well and good when we have prokaryotic DNA and it's really simple and we don't need all these factors. Let's talk about eukaryotic gene expression. So the phenotype of a cell or an organism is determined by a combination of genes that are expressed at levels that they are expressed. What? That doesn't make any sense. Essentially, the phenotype is determined by multiple genes that come together and are expressed at different levels. We saw that with non-Mendelian genetics, with like the codominance and the incomplete dominance. It's multiple genes that are determining a phenotype. Differences in cell types, we call that differential gene expression, right? So the expression, the gene expression that's found in my skin cells are different than the gene expression that's found in my liver cells, right? They serve different functions, they need different proteins, their gene expression is different. So we're regulated at different stages. There's a lot of different regulations that can happen. First is chromatin structure. So you can see chromatin all the way up here. All right, that's kind of the first step of it all. So we could have DNA that's more tightly wound, it's less accessible for transcription. That happens because before we get into this, you all know that every cell in your body has the exact same DNA. The only thing that makes a liver cell different from a skin cell, different from a colon cell, different from is regulation of genes. So if I essentially took, theoretically, this is what got me so interested in this stuff, I could theoretically take a skin cell, get off all of the operons, take off all of the repressors that make it other things, put on the inducers and the different sequences and the different regulatory things, and make it something else. It's really kind of cool. Anyway, so when I'm a skin cell, the chromatin might wind up a little more tightly in the areas that code for a liver cell, right? So the chromatin structure itself allows these genes to be regulated and turned on and off. How can it be modified? Well, we have histone acetylation, right? So it adds acetyl groups to histones, essentially being able to sort of loosen the histones. Remember, the DNA is wound around these proteins called histones to have that more compact structure. It could also add methylation, right? So it adds a methyl group to DNA and causes it to condense again. So acetylation loosens it, methylation tightens it up. Pause here, take notes. Cool. Another one's epigenetic inheritance. So it doesn't alter the sequence of nucleotides. Obviously, if we take the chromatin and we twist it or loosen it, it doesn't change the actual sequence. But it could be heritable to future generations. So modifications can be reversed, unlike mutations. This explains why one identical twin may inherit a disease that the other one does not. So epigenetic inheritance is just this idea that multiple different modifications and multiple different regulation sequences actually cause these differences, these heritable differences to future generations. Transcription initiation. So once chromatin modifications allow the DNA to be more accessible, specific transcription factors can bind. So remember, this is just going over the different ways that genes can be expressed and regulated. So one is being able to coil and supercoil the chromatin, right? Open DNA, that sort of thing. Now we're looking into specific DNA modifications, so things like this pre-mRNA and the exons and the introns and the cap and the tail. We're looking at specific non-coding sections of DNA. These are binding sites, right? So there are parts of your DNA that don't code for anything, right? This is how we sort of express eukaryotic genes. So it can be increased and decreased by binding of these activators and repressors to control these elements. Remember, this whole topic is about control of these genes, control of these gene expressions. Produce more protein over here, less protein over there. Right, so these non-coding sections that serve as binding sites can be different from one to the next, and that could cause differences in these elements, in these that make it one cell to another cell. These different proteins that have to be made. RNA processing, right? Alternative splicing of RNA. Being able to take this pre-RNA and mixing it up to be able to produce different proteins. Translation initiation, right? It could be activated or repressed by initiation factors. MicroRNAs and small interfering RNAs combine to mRNA to degrade it or block it during translation. Don't worry too much about that. MicroRNAs are something you'll find in college. But it's essentially, you should know the process, that there are these other pieces of RNA that degrade RNA, right? So you have an RNA molecule here, you see degraded mRNA. When we're done, when the cell's done with mRNA, we're done with it, we just degrade it. Sorry, pause here. So I do just want to point out that all of these were examples. Let's just go back to the main header here. How eukaryotic gene expression is regulated at different stages. So we went over the chromatin stage, the genetic stage of the genes itself, how we transcribe it, how the RNA is processed. Sorry, we went through a lot of steps really fast, so I just wanted to make sure that we stayed with like the overall conception here. Cool. Eukaryotic development. So during embryonic development, cell division occurs. We call that differentiation, right? When some cells become specialized. This is these are like where this is like past the stage of stem cells, right? Morphogenesis is the terminology that we use to describe the physical process that gives an organism its shape. And right down here, you can see here's 2 cells, then there's 4 cells, and there's 8 cells, and 16 cells. At this point in development, we call it a mortula, then a blastocyst, and then that goes on um, to grow and divide to be a number of different cells. But yeah, isn't it weird that we kind of start with 2 cells and we end up with like a bunch of different ones? This is kind of how that happens. Pause here if you need to take notes. So, how do cells differentiate during early development? There are substances in the maternal egg that can influence cells. There's cell to cell signaling, right, that can cause a change in gene expression. Both of them influence the pattern formation itself. So essentially, the first thing in human development that develops is spinal cord. It's like a spinal node, right? So everything, all the cells kind of move up there and they differentiate to form different patterns that go on to form different functions, et cetera, et cetera. It's really kind of a cool thing. Embryonic development is fascinating. It's a body plan for the organism. So, like, the cells sort of move to a point that everything is in place before they start growing and dividing and genetically differentiating from one another. It's really interesting. Um, Homeotic genes map out body structures. Don't worry too much about that either. Just know that there are genes that they code for you should go here, you should go there, right? During embryonic development in eukaryotic cells, right? It's really cool. So there are these genes that are only active during development as well, to kind of form that body plan before things um, start growing and dividing. This is also how people can end up with like the mirror image of your organs, right? I forget the exact name of it, but it's like normally your heart is slightly to the left of your chest. These folks have it on the right. Um, if your body plan, quote unquote, for your organism gets messed up in these early stages, things can go in weird directions. Cool. Pause if you gotta take notes. All right. As cells differentiate, apoptosis, programmed cell death, plays a critical role. It allows structures to take their form. So, for example, when we are in the womb, we have webbed hands and feet. Apoptosis goes through and kind of breaks all of those skin cells and all those cells that would be that are like the webs in between. Cool. That's it. 23 minutes, not too bad. I'll see you tomorrow.